Welcome. I'm Richard Shyness. I'm the Dean of the Dietrich College of Humanities and Social Sciences at Carnegie Mellon and a part of this fantastic Center for Causal Discovery. It's a pleasure to have Karen Sachs here today to give a distinguished lecture series. Let me just give you a brief bio of Karen, and then I'll just let her take it away. So Karen is now a research scientist at the Stanford School of Medicine. She did her PhD under Dr. Doug Laufenberger in bioengineering department at MIT. And during that time, she actually worked and introduced all kinds of interesting techniques on getting single-cell data to track signaling pathways of all different types, in particular for doing causal inference. She spent a bunch of time working in combining single-cell data, machine learning, applying the algorithms that many of us are familiar with and working on ourselves. And she came to Stanford University School of Medicine as a Leukemia and Lymphoma Society postdoctoral fellow, where she worked under the supervision of Dr. Gary Nolan, who is a world leader in single-cell proteomics. And her application areas include cancer, especially blood cancers, drug response, and immunology. Pleasure to introduce and have Karen Sachs.
Thanks a lot. Hey everybody. So I was not entirely sure who my audience would be today, and the last time I was in this area, I was talking to a strictly causality community. And so I put in a bunch of stuff on the biology domain and a bunch of stuff on the measurement approach. It sounds like the biology is covered. You guys don't need to understand what protein is that right? Yeah? Everybody's familiar with the biology, so we'll skip those slides. I do talk a little bit about the measurement technology because if you're interested in working in single-cell proteomics, the measurement technology is nuanced. The nuances are important, and so that might be interesting to some of you. If not, we can skip some of those slides. So these are 3 cells. They're undergoing apoptosis. And we've measured 3, we've labeled 3 of the proteins in them. And my focus has been on what can we learn when we measure things in individual cells. And I'll get back to it later, but I just want to show you how pretty they look. So cells respond to their environment. You guys know that. They get some kind of a signal from the outside. They make a decision. And that decision is based on a pathway inside.
Just a little delay.
Exactly. Okay. And of course, this breaks down in disease state. This is why we want to measure these. This is why we want to model these signaling pathways. So for example, in cancer, the cell might decide to proliferate incessantly and cause a tumor, etc. Okay? This is review for everybody? Anyone not know signaling? No? Okay. One person? Everybody's good. Okay. So my motive, the overview of sort of where I would take these techniques, if I had a good causality technique, is to do something like this, where I'm asking what breaks down in the disease state, or can I characterize this particular patient's tumor, or can I characterize a particular drug, because I want to know where the drug hits in the network, so that I know how to take a disease network and bring it back to something that looks more like normal. So this is the motivating idea behind my work. We want to map these signaling pathways so that we can have clinical impact. So here's an overview of today's talk. First, I'm going to talk a little bit about where does the data come from and describe the technology. I worked a fair bit on the technology, and like I said, I think it might be interesting to some of you in case you're interested in trying out that domain. What kind of causal connections happen in this domain? So that sounds a little trivial, but I think that the way the signaling pathways work and the things that we're able to measure combine to cause certain issues, and we'll come to that. And finally, what is needed for causal learning? So what are the outstanding challenges in this particular field as I see them? And that's kind of a discussion I'm more than happy and in fact eager to hear your guys' input. Maybe you guys can teach me something there. So first of all, where does the data come from? So I think you guys know this. In general, in biology, it's always been the case that you take the tumor sample and in 90% of the labs, you put them into a blender. And then you measure the proteins, let's say with a Western blot. Is everybody familiar with this? This is how biology usually works. Lysates, and of course that gives you a vector. And with a vector, I can't do any kind of causal inference on one tumor sample. I can take a cohort of 100 patients, but I can't do much with this kind of analysis in one patient. So what we're going to move into is a paradigm of single-cell biology. And why do we want to do single-cell biology? From the biological perspective, it's because a lot of the tissues that we look at are mixed up, many different cells together. They're heterogeneous. And for example, in cancer, you have a complex system with lots of different types of cells involved. So single-cell measurements are important from the biological perspective. From the statistical perspective, if you look at individual cells that are measured in high throughput, then you have a high throughput data source. And now you can characterize individual cells, and you can get a large number of data points if you consider each cell as an observation of your system. And this gives you thousands of data points. This realization was really the only real contribution of my entire PhD, was to say, oh, wait, single cell data is actually, if you look at every cell as an observation of the system, then you have a ton of data. No one had done that. No one had said, oh, look, let's look at single cell data and say, oh, I have a huge population of data points. This is amazing. I can learn things from this. I can learn the joint probability distribution. I can try to do causal analysis. This realization was really the rest of the time I spent at MIT. I shouldn't have bothered. No, I'm joking. But this is the main point. Okay? And this realization says, hey, why don't we use this single cell data? Why don't we do statistical inference on it? Let's see what we can do with it. Because when you measure single cell data, of course, you get this kind of a big matrix instead of the vector. So that's the whole motivating idea behind what we're doing. And so the idea is to take this kind of data and to learn the types of signaling pathways that we talked about previously. So in my PhD, I said, oh, this is a great idea. Let's try this. And we took some single cell data that had been perturbed in various ways. It was just normal human T cells. They were put into this multi-well format, and they were perturbed with different specific inhibitors. We hope they were specific. We got a large data set with thousands of data points and a bunch of specific inhibitions. And all I did here was run Bayesian network structure learning. Fairly straightforward. There were a few ways where I had to plug in the algorithm into this particular domain, but it was reasonably straightforward. And what we got was a model that looked a lot like what you would get when you open a textbook and look up T-cell signaling. So that was really encouraging. There was one connection that we validated with siRNA, et cetera. And we looked at this and we said, this looks great. Is the problem finished? And what we'll realize a little later is that no, the problem is not finished. But for now, let's talk about the single cell data. Just let's introduce the technology. So here's the multi-well format that I showed before. And here are 4 cells that I've zoomed in on. The original technology was called flow cytometry. And in the original technology, the antibodies, which are tagging the proteins, are attached to fluorescent dyes. And we're going to quantify the amount of fluorescence. So here are 4 individual cells, and you can see that this cell has a lot of whatever's tagged in green, and this cell has a lot of whatever's tagged in blue, the different proteins. And in the flow cytometer, a laser and then the detector is used to quantify the amount of fluorescence in every individual cell. The problem with flow cytometry was that it was dimensionality limited. And so what we're trying instead is to use mass cytometry, which is exactly flow cytometry, more or less, except that the antibodies are attached to these elemental tags instead. And we'll talk about this technology a little bit more in depth. But the concept is extremely straightforward. Switch out the fluorescent probes with an elemental tag and throw the cell into a mass spec. And this is what the first version looked like. It was this beautiful shade of orange. The newer ones are a beautiful deeper orange. And here's a little bit more background on the technology. So this was fluorescence, and this is how the fluorescent dyes overlap with each other. And you can see that when you try to separate these, you're going to have some signal-to-noise problems, especially if you have a large signal that overlaps with a small magnitude signal. The noise is going to really interfere with the accuracy of the measurements that you record. And so the mass spec spectrum looks much, much better. And so you can throw many more parameters into the spectrum. So the antibody labels are isotopes of elements. We're going to get them from the lanthanide series over here. And each one is actually, that you see here, is actually more than one isotope. So you're actually able to use each individual isotope as a label, because the mass spec has a very easy time saying that I have this much of mass 152 and this much of mass 153. We're still dimensionality limited. So right now we can do about 40 parameters with this type of approach, which if you come from the flow cytometry world, you get up and you cheer with joy. However, 5 minutes later you say, 40 parameters is not enough. In signaling, we're probably dealing with hundreds of parameters. We have antibodies for hundreds of parameters. We would really like to be able to measure more. If you're interested, I'm working on the last year or so, I've been working on a method for increasing this dimensionality. Talk to me offline. It's not causality based. I didn't include it in the talk. Although if we have time at the end, I could. Okay, so a little bit more details about the technology. Here is an antibody, and this is how the antibody is tagged. It's attached to a polymer, and every polymer is attached to multiple ones of these metal ions. And this is a process. First you chelate the metal, and then you conjugate the antibody to the polymer. So it's a chemical process. Now, this is important. Why? Because when you perform these conjugations, I've actually performed them myself. Sometimes they let me into the lab. When you perform these conjugations, let's say I'm going to conjugate protein whatever, ERK, and then you're going to conjugate protein ERK. The efficiency of our process might be different. Okay? And the important thing to understand here is that you cannot really compare one experiment to another necessarily. So the actual magnitudes cannot be compared across experiments, okay? Unless you take special measures. But it's important to understand that there's a lot of stochasticity in this process. There's the stochasticity of how strongly does the antibody bind the protein. That's going to differ from one protein to another, one antibody to another, especially if they're polyclonal. So if they're polyclonal, they're going to be variable from the onset. And this process itself has a lot of stochasticity. Okay? How many of these, whether you do a good job, basically, and saturate everything or not.
So are these made in the lab or are they commercial?
Both. You can buy a lot of them from DVS, which was acquired by Fluidigm. You can buy them, and many people do. We do a lot of conjugations ourselves. If you want to really fill up the spectrum, you might end up doing conjugations. You get more flexibility that way. It's cheaper, I think. Okay, so here's an overview of our workflow. You start by taking your cells, they're usually in a test tube or a plate or something, and you stimulate them. And the reason that we stimulate them, maybe you guys already appreciate this, if the cells are just hanging around and I want to characterize, I don't know, this cancer patient who responded to chemo and this other cancer patient who didn't respond to chemo, I might not see anything. But if I stimulate them, it's like if I came up to you and I poked each one of you, some of you would laugh and some of you would scream and some of you would throw something at me. And I would learn more about your internal state by stimulating you. Same thing with the cells. You can look up a beautiful paper from the lab from before I joined, Jonathan Irish's cell paper in 2004, where they showed that the cancer samples and the normal samples kind of look the same. When you stimulate them, they look completely different. And suddenly you can see these are the cancer patients who are in big trouble, and these are the cancer patients who are going to make it. You just stimulate, and suddenly you can see the internal state. So that's the idea there. Now we're going to cross-link the proteins. Why do we cross-link the proteins? Because we need the guts of the cells not to come out when we make the holes in the membrane. And here we are making the holes in the membrane. We've permeabilized the cell membrane. Why do we do that? Because antibodies are big proteins, and we need them to go in and bind to those signaling proteins, okay? So we permeabilize the cell membrane, and now we stain with isotope-tagged antibodies. Which antibodies do we use? Depends which proteins we want to look at. How do we pick those? You know, that's magic. I mean, you either know something about your domain, or you've done some kind of preliminary analysis using gene expression. I don't know. Usually you have your favorite proteins that you think might be a good idea, or maybe you do a pilot experiment to begin with, or something like that. But also to pick your stim, you might do a pilot experiment. You might just kind of close your eyes and guess. So now the cells go through this nebulizer, and they go into this very hot plasma that's as hot as the surface of the sun. And the cell is completely annihilated, and all that's left are these ionized metal tags. And they make this big cloud. And the cloud of ions goes into the mass spec. And the mass spec quantifies how much you have from each mass. So here's the ion cloud belonging to cell 1, and the mass spec said, there was this much of this mass and this much of that mass, and so on. And then at the end of the day, just like in regular flow cytometry, you can make these kind of scatter plots and say, oh, how much did I have in this cell of CD45RA, and how much did I have in this cell of CD8, and so on, different proteins. Here's a little movie of this happening. So here's the cell. It comes out of the test tube. It's going to go into the nebulizer, and then it's shot through this time-of-flight mass spec. And the time-of-flight mass spec uses the amount of time that it takes the ion to fly across some chamber or something to decide which mass that was. And this is what it looks like. So the ion cloud goes through the mass spec, and the mass spec doesn't know when there's a cell coming. So you want to put in a dilute concentration of cells. And it's pushing the ions down, constantly, just pushing them down, push, 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 push. And some of the pushes don't have very much going on when there's no cell coming through. And sometimes when it pushes down, it sees a particular mass. This is mass 131, for example, and so on. And this is the raw data. This is what it comes out looking like. And then there's an algorithm that says, did we detect a cell? A way of saying, have we found a cell here? And then there's an algorithm that says, okay, let's find the boundaries of this cell. This is where the cell started, and this is where the cell ended. That algorithm is proprietary, DBS black boxed it. You can hack it. You can make your own algorithm, and we have. If you do, it might make you concerned. It's made some people concerned. So you could, if you want, you can go back to these IMD files, they're called, and you can make your own cell detection algorithm, and you can start at that basic level if you want to. We've tried it a little bit in the lab.
That's another question. So, you can use multi-dose, of course, without that.
It's a very different experiment, yeah.
Can you use that for essentially standardizing your antibody state? So like an unlabeled sample with a labeled sample?
I'm not sure. I don't know a whole lot about mass spec that's not tagged with antibodies. But I do know that when you do that, when you do untagged mass spec, regular mass spec, it's a whole other Pandora's box. And you have to worry about things like different abundance in the proteins, and there's a bias in which ones you see.
That's exactly the point, that you can standardize certain about the protein, right? You use an antibody, uses a non-antibody one, and you potentially standardize.
Yeah, maybe. Yeah, except you can't say anything about the abundance that you actually see. So if I label, I'm doing this experiment, and I do two proteins, protein X and protein Y, and protein X gives me a histogram with a mean of 1,000, and protein Y gives me a histogram with a mean of 100. What can I say about the relative abundance of X and Y? Nothing. Because one might have a really weak binding antibody, and one might have a really strong binding antibody. Even worse, one might have an antibody that also has background binding to other stuff. I can say absolutely nothing about the relative abundance, okay? I mean, if you tried 20 different clones of antibodies and protein X never gives you a high signal, then you might start to suspect that protein X isn't very highly expressed in the cell. But as a rule of thumb, you can't say anything about the difference, okay? So
strictly covariance.
yeah, but you can compare. So if if I measure my blood and your blood, I can say, oh, you have more protein X than I do, and I have more protein Y. You can say that, especially if you measure them in the same experiment using something called barcoding, which I'm not going to describe here, although I could if you want. Okay, so these are the different cells. You pick them out and quantify. You pick them out and you quantify. Sorry, this delay is driving me crazy. Maybe I'll use this directly. This is also delayed. Okay. It's my computer.
Maybe it's the.
Oh, maybe it's because it's on mine. Oh, okay, sorry. Okay, I'll try to be. I'll just be patient. So these are the individual cells, and then you quantify and you can see that this cell has CD2, and this cell has CD19. So this is a B cell, and this is, I think, a T cell or something. Yeah, a T lymphocyte. Okay, so raw data. The last thing that I want to point out is that the machine can be finicky. So here is bead data that's across different instruments on different days. Now these are beads. The beads are pretty standardized. They should all kind of give you a straight line. And one thing you should notice is that they don't give you a straight line. The data is smoothed. To begin with, the data is smooth. And even though it's smooth, it's still a little bumpy. So what happened? I don't know, the plasma flame got diverted because somebody walked into the room. I don't know what happened. But when you actually look at the data, it's very, very bumpy on a very local level. After you smooth it, you still see some things that are concerning. One thing that I find particularly concerning is that here, the blue bead is higher than the red bead, and here they've switched. So it's something to be concerned about. There's a nice paper out of the lab where a bead normalization technique is suggested, and that really helps standardize things a lot. Okay, but these are just things that I want you to be aware of if you use this kind of data. Of course, I'm a big fan of this kind of data, don't get me wrong, but I think it's important to walk in and understand what are some of the nuances that you need to think about. Okay, so signaling 101 I'm going to skip. Oh, except I do want to say something. So here's signaling. A gets phosphorylated, it causes B to be phosphorylated, and then C, there's a cellular response. It's read out. The important thing is this. The antibodies that we have are generally against the phosphorylated form of the antibody. Of the protein, of the signaling protein. We almost never measure these guys. The unphosphorylated guys, they almost never get any attention from us. Those antibodies are also harder to validate because you need a cell, in order to see that it's on, you also need to see that it's on. Where are you going to get a cell that has no work, right? You have to make a knockout cell line or something. So that's complicated. These are the guys that we measure. Keep that in mind. When we think about signaling, I only think about the action players, these phosphorylated guys. But sometimes we have to worry about the fact that we're completely ignoring their predecessors over here. Okay, so what is needed for causal learning? This is my personal view of what challenges remain in causal learning in the field. And some of it is quite domain specific and some of it is less. You guys are causal learning people. I'm sure you know causal learning better than I do. I would love to see you guys working on this. If any of this strikes your interest and you want to talk to me about it more in detail, by all means, I would love to see these problems solved. I've worked on some of them, but not all of them. So here's what I got using Bayesian networks. And it looks really good, pretty good. But first of all, we have quite a few perturbations. And second of all, we ignored things like cycles and things like that. So let's see, even though, I think a lot of people read this paper way back when I was a grad student, and they said, the problem is solved. And I think that's very not true. So I think people from the field probably understood that. But let's see why that's not true. Okay, so here are the things that I still see. Here's an overview. And those of you with background probably read the paper back then and said, wait a minute, what about these things? What about cycles? Absolutely, the cycles problem has not, at least in Bayesian networks, has not been solved. There's a CCD. So when you pick the graphical model that best fits the data, and you optimize that Bayesian score, then you need to parameterize it somehow. You need to express the probability of the model given the data in some way. Whether you use a linear Gaussian, you want to use a multinomial, which is what I use, you got to do something. And when you do that, you're going to change your answer. I can tell you, having tried a whole bunch of these, that the answer is specific. So the CPDs are an issue. The cycles are an issue. Other things. Yeah, I'm just kidding. So other things that I worry about a lot are hidden variables. And one thing that I'm going to mention is also the dynamics of the system, because we're taking a snapshot of the cells as they're moving. Okay, so let's talk just briefly about the CPDs. This is a quick analysis that I did using just multinomials. Now, I like multinomials because they can be arbitrarily complex. The relationship that they express can be arbitrarily complex. But of course, you have to discretize for them. And when you discretize, where should you discretize, and how many bins should you discretize to, and how should you perform your discretization? I don't have answers to any of these questions. So I just did a quick little analysis where I said, okay, well, let's look at this was a data set in some kind of human PBMC. And I said, let's see how much things change. And the take home from this slide is simply that some edges are super robust. When you switch from 20 bins to 3 bins, some edges are super robust, and some edges are super not robust. Which one is true? Which one is capturing ground truth? I don't know. So that's something that we need to think about. I wish it would give me a preview of my next slide so that I would know what I need to start talking to you guys about. So the CPDs need expressive power. This is just a figure that I stole out of Smita Krishnaswamy's very nice science paper with Donna Pereira last year. And in this paper, they took and they smoothed their binning here. They're doing the conditional density based on the x-axis that they're showing in the y-axis. So this is like highly smooth. The data doesn't look nearly this clean, but because it's highly smooth, it looks nice. And you can see, just wanted to show you that some relationships look a little bit linear. Some of them look kind of more sigmoid. Some of them are more discontinuous. There's like a population here at the low and a population here at the high. What kind of CPD would best parameterize that? What would capture it? Of course, a multinomial could capture it, right? Given sufficient bins, a multinomial will capture kind of almost anything, right? That's why I like them. But at the same time, if you think about it, especially if you start going into a higher number of bins, you're going to have a bunch of bins that have like no cells in them at all, and then you're only going based on your prior. That doesn't quite feel right either, right? I was speaking with Joris Moy, who some of you may know, in Amsterdam this summer, and he was talking about how using Gaussian processes as a more flexible CPDs. I've done very little work on this. I think it's a really, really important problem. And I can tell you that it's really frustrating to run your algorithm and to change the underlying CPD and get a completely different answer. To say, well, I don't know. What do I trust? I don't know. So I think this is actually a really important problem. Okay, so what about hidden variables? Hidden variables are a huge, 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 huge issue, especially when you run the score-based types of Bayesian network that I run. They don't handle hidden variables very, very effectively. And so you might get a lot of inferences that don't make sense, and you show them to biologists, and of course the biologist says, this isn't right. And you're like, well, of course this is modulo hidden variables and so on, but you can't be a good hypothesis generator if you don't worry about the hidden variables. So I think these are a huge issue in the domain. And the first thing I'm going to talk about with this, I kind of have two things for you guys to think about in the hidden variables. The first thing I want to talk about is how to use data integration. That is the obvious answer from my perspective. Data integration will solve our problem. So if we can get to the slide. Here is a depiction of human immune system. And I'm going to pick two populations, and I'm going to just run Bayesian network structure learning on them and compare the structures that I get. And when I did that, I got this structure for memory T helper cells and this structure for the naive T helper cells. And let's just focus on one edge that was different between them. They had some similarities and some differences. Let's pick out an edge that was different. This edge here between STAT1 and CREB. And the reason I pick on this edge between STAT1 and CREB is that this data set was run at 30 minutes. And if any of you know STAT1, you know that there's no way that STAT1 could have any influence on anybody in 30 minutes. It's just too soon. STAT1 works by modulating transcription, not by modulating signaling. So I know this is not a causal edge. And if you show this to your biologist friend, they're going to say, your algorithm doesn't work. So what's probably going on? How can I figure out what's probably going on? Again, the obvious answer to me is to use data integration. Oh, by the way, you look and you say, is my algorithm accurate? Is there actually a statistical dependence? Of course, the answer is yes. And you're not surprised because you know that your algorithm works. So can we figure out what's actually causing this edge? So this is a method that I tried for adding in the hidden variables using just data integration. Going to the prior knowledge literature and asking, can I figure out what's causing this edge between STAT1 and CREB? So I have a statistical inference. Now I'm going to extract potential underlying molecular paths. I'm going to say, what could have connected STAT1 to CREB? In this case, it's an easy case because I know that STAT1 is not phosphorylating CREB. STAT1 is not a kinase. But in general, I have to ask the question, is there a mechanism? Is there a directed path that goes from STAT1 to CREB? Or perhaps did I get my arrow wrong? Is there a directed path between CREB and STAT? Or is there a hidden variable that connects them both? So I just look in the literature and I build backwards. And I say, can I find any one of these three paths? In this case, I know these two are wrong because these guys are not kinases. So I know to narrow it down to here. But in the general case, I don't know. And I just look in the literature. I go to the databases. You can even go to something like ScanSite, which makes in silico predictions. And you ask, what are the putative paths? Now, if you get just one putative path, there's your hypothesis. You're done. If you get 0, I guess you're stuck. You can go to lower confidence types of databases. If you get multiple possible paths, then you have to rank and select the most probable paths. And in my case, I had multiple probable paths. And so I went to the gene expression, and I said, which ones include molecules that are differentially expressed between the naive and the memory T cells?
Why do you only limit it to having one path? There could be two.
Oh, or there could be, but sometimes you get 1,000, and you want to be able to sift through them and rank them.
No, I mean, unless I'm misunderstanding, you have three possibilities. Path in one direction, a path in the other, a common cause.
Oh, you're saying could be both of these.
Could be a combination.
Oh, absolutely, absolutely. And so in this particular case, because this is not a kinase, there was no point in asking these two questions.
Right, but
in general case, absolutely. You run this, and you run this. And then that gives you the compendium of possible hypotheses, okay? And because it's not that useful, just like it's not useful to hand the biologist a hairball, it's not useful to hand the biologist 1,000 different hypotheses. So you want to rank them, and that's what this piece is about, okay? So in this case, I went to the gene expression, and I said, is there a path that stands out because it includes a delta expressed component? And there was. Well, I had to go to mice. This is human signaling, but mouse gene expression. But there was in this case. And so we were able to generate the following hypothesis, that the edge between STAT and CREB is probably because of a hidden path that connects them both, that goes via these molecules, which were delta expressed in the two. They were highly expressed in one of the cell types and low in the other, I forget which. And the implication here, so we're sort of able to complete our story, with the Bayesian network path, we look at the differences between the two types of cells, and we're able to make some kind of conclusion. In this case, our hypothesis is that this branch that you guys are seeing here, that my data integrator picked out, is more active in naive, and it's not active in the memory cell. So you can complete that path and go back and try to validate that in the lab. So I want to say something else about these hidden variables. There's a special case that's that I found pretty interesting that showed up in this particular domain. And to talk about it, we're first going to talk very, very briefly about the cycles. We're going to talk about them very briefly for two reasons. First of all, I already talked about them when I was here 2 years ago. And second of all, it's published. But I'm going to mention them because they're the segue into where I discovered this special case of hidden variables that I found intriguing. So me and one of my favorite collaborators, Salman Itani, we said, let's try to think about cyclic structure learning. Now, I know that SCMs already do cyclic structure learning, but they are limited to linear, or we wanted to try this other approach. And so the approach that we suggest, and I'm only going to show one slide on it, you can look it up if you're interested, or talk to me offline, but the approach that we suggest is to say, in our domain, when we inhibit, usually what we do is activity inhibition. We don't take mech out of the system. We just don't let mech do anything. Okay, it's paralyzed. It's there, but its hands are tied. So when you measure the abundance of mech, you still see lots of mech. But when you look at the child is not active. Activity perturbation. When you do an activity perturbation, this is real data of an activity perturbation of PKC. And when I look at PKC under stim alone, it's substantially higher than when I inhibit PKC. Why? Why? All I've done is tied PKC's hands. And now I'm measuring the abundance of PKC, and I see that it's gone way down. And so we said, if we see that, we're going to conclude that there's a cycle from PKC back to PKC. The first step, we detect the cycles. And the next thing we're going to do, and I'm not going to go into this, but the next thing we're going to, just conceptually, is we're going to use these small molecules to break open the cycle. And now that we've broken them open, we can use regular patient structure. And then we have to close the cycle using some statistical tests. So that's the idea. And in order to test this algorithm, and in general, I think when you're working in causal discovery or any algorithm where you want an answer and you want to know is my answer correct, it's really nice to have very highly reliable synthetic data. And that's what we were trying to do here. So we went to some friends at Merrimack Pharmaceuticals. These are people who came out of Doug Leffenberger's lab and neighboring labs. And we said, hey, these guys have these dynamical systems models, ordinary differential equations, where they're trying to mimic a signaling pathway. And they're trying really hard because if you have any idea how much a clinical trial costs, they pick their drugs based on these clinical trials. And by the way, they just got good, not based on this model, but a similar model from the same company. They just got, I think, good results in their phase 2 clinical trial or something. I'm forgetting the details. But they are extremely motivated to make these models as accurate as humanly possible. And they validate them very, very rigorously. So this is going to be my idea of the best case situation where you're going to get data that's very, very similar to what you would get in a lab, but you have a ground truth. Okay, so now let's focus in on just this top part of the model. When we did the analysis for this top part of the model, this is the ground truth, and this is what we found. This is the ground truth, and this is what we found. And you see that there's an edge here between IGFR and PI3K. And this was a very strong edge. And it was super persistent. And I literally called up Jonathan, and I said, Jonathan, there's a mistake in the model. You forgot to tell me about an equation that links IGFR to PI3K. And he said, no, there isn't. I know my model. I'll send you the code if you want me to. I was really, really convinced that there had to be something going on. There was a very, very strong and persistent relationship here. But it's not written into the equations. This reflects what was written into the equations. And I really scratched my head and could not understand what was going on. And now I'm going to remind you guys of something. That we talked about this being signaling, but these guys are latent variables. Don't look at them. So what actually happens in signaling? And what actually, let's zoom in and say what actually happens in signaling. And I'm going to use a slightly different example, but the intuition is the same. Let's say I have A phosphorylates B and also phosphorylates C. Okay, so what actually happens? Let's look at the details of the model. So A becomes phosphorylated A. I don't know how. Let's not worry about that. Let's say a ligand binds or something. And it's going to form a complex with inactive B. That's the first step. And then it's going to phosphorylate B and release it. I think I probably forgot also there's another complex here, a phospho A bound to phospho B before phospho A is released, and phospho B is also released. And what happens with phospho A, or with C? The same thing. Phospho A binds to inactive C, and then you have another complex that I'm not depicting, a phospho A bound to phospho C, and then they're both released. So I think you guys probably see where I'm going with this. If I have a limiting amount of phospho A, what's going to happen to these apparent siblings, B and C? A causal edge will appear between them. Let's say B is more abundant. What's going to happen is that B, more abundant B over here, is going to suck up all the phospho A. And phospho A is going to be sequestered. At the limit of insufficient A and say infinite B, phospho C will never get phosphorylated. And so this is just a latent variable. We all know latent variable, but to me it's an interesting domain-specific example because we scratched our heads for a long time over this to understand, what's going on and why do we have this edge. But at the end of the day, it's just a simple latent variable issue. But it's something to be aware of because we don't even bother depicting these guys when we draw the models. Who cares? They're not doing anything. But they're actually quite important. And in fact, if you guys know Stas Schwarzman, who's a dynamical system model guy who works at Princeton, he does amazing work in Drosophila. This approach where, in essence, B, if B is more abundant, B ends up affecting A, this approach is actually critical in Drosophila development. He showed it one day. He discovered this mechanism, and I said, isn't that interesting? It doesn't matter how much phospho A you have. It matters in general how much A. But actually, you can see that B, the sibling here, is affecting its other sibling, even though it's not phosphorylated. Although, of course, if you have a lot of phospho B, it's a good proxy for there must have been a lot of B in order to get a lot of phospho B. Okay, so even though these guys are always hidden variables to us, and this complex is a hidden variable too, when you have your antibody for phospho A, is it measuring this phospho A or is it measuring this phospho A? Probably it's measuring both. It might not, because of steric hindrance, it might not measure phospho A that's bound to its target. But I think in most cases, you're going to just get the total amount of phospho A.
So you have your A, B, and C to the lower right. You're actually measuring the phospho A, the phospho B, and the phospho C, right?
This is my ground truth model. And when we draw a ground truth model, we don't bother with these guys.
Right. But
We only draw the action players.
What you're measuring is the phospho A, the phospho B, and the phospho C.
Yeah, that's why you have this problem.
But conditioned on what you measure, which is phospho A, purple, isn't it true that phospho B and phospho C are independent given that phospho A?
That's what I would think. You would think that. But here's why they're not.
Okay, how is that?
Because of this complex right here.
And how does that complex deconnect them?
Because when phospho A is here, it can't do any phosphorylation. So actually, if you think about it very strictly, B is a parent of phospho A. Because here it is. Never mind B, this complex of being bound with B or bound with phospho B, that's the complex I didn't depict. It is a parent of phospho A. So if you have, it's going to affect how much phospho A there is in the system.
Then it's the amount you can measure possibly, right? That's the idea.
What's that?
The amount you could measure possibly is limited by how much B there is.
Not the amount you could measure.
Well, it could be seen. I mean, so it sounds like when you have a certain amount of phosphor A, right, going out to the B and C, right? If there's only a certain amount of it, and B soaks up some of it.
Yeah.
Then that's not available to B.
Right. So if A is infinite, I wanted to put up a picture of it. Did you guys see the headline that was going around in some town? Rejected solar panels because they were afraid that it would suck up all the sun. So this is this example, right? No, those people are stupid because the sun is not a limited resource in that sense, right? The fact that the solar panels are sucking up the sun will not keep the crops from sucking up the sun because the sun is not limited. But let's say they were right. Let's say these people's intuition was correct, and there was only 1 million photons coming down to planet Earth, and those solar panels were able to suck those photons towards them. If I put a huge number of solar panels, they're right, their crops will not get sun, right? That's what's going on here. This A is not the sun. If it is, this problem will not arise. That's why I only see one edge like that in my whole model. There was one edge like that. There were potential for a lot of edges, but there was only one. Because only in that situation did we have not the sun, but just a limited number of photons coming down into planet Earth, right? So only because A is limited. If A is infinite, this won't happen because I don't care that B is affecting the amount of phospho-A available because the A is unlimited. But if A is limited, if A is on the order of B plus C, then here I have B sucking up A, and B is the parent of phospho-A. And so B ends up, whoever is bigger, ends up affecting the other. Although if they're within the same order of magnitude or whatever, if they're in the same range, then of course they'll affect each other. B will affect C and C will also affect B. But if we're assuming that C is much smaller than B and that A is limited, you'll get this effect only under those conditions. And that's also something very interesting because if you're working in this domain, it's something to think about. What happens, did I make a slide for this? First of all, this will not be revealed by an activity inhibitor. If I block phospho-B, what's going to happen to C? Absolutely nothing. Why? Because it's not the kinase activity of B that matters here. It's the binding activity. If I instead block the binding of B such that it cannot form this complex, then I will take away this edge. It's very nuanced.
Oh, why would that be? You have more available to bind with C now.
Yeah, so B would no longer affect C if it wasn't sucking up A. Oh, you're saying if I block B, I'll see that C goes up.
Yeah.
That's true. That's
You
true.
have, yeah.
Yeah, that's absolutely
You
true.
took the direction.
Yeah. Well, I guess I was trying to say,
The sun.
if I block the activity of B, then I will not affect this edge at all. In order to affect this edge, in order to see the effect of my perturbation, I need to affect the binding of B to A. Okay? So it's a different kind of perturbation that I would need to do. It's very nuanced, but it's something that's important to understand. And I think if we're building these models for the clinic, we have some kid whose leukemia failed the standard of care, we profile their tumor, we get this kind of causal edge, and we think, oh, well, maybe I should use an activity inhibitor against B in this kid. Well, no, you shouldn't. So it's important to think about this. Is there a statistical test that will reveal to me when I have this kind of regular causality where phospho-A affects phospho-B, and when I have this other type of causality? Can I look at the domains of the magnitude of A versus the magnitudes of B and C to try to discover when I have this type of effect? When I'm thinking about discovering hidden variables, can I ask the question in this particular domain when I know that all of my unphosphorylated forms are all latent variables? Those are solidly established. I know they're there. I know I haven't measured them. I know they're doing something. I don't know if they're doing something important. Is what they're doing important to causality? Can I formulate my latent variable discovery algorithms in such a way that focuses on this and asks this question, is this going to happen? Because we know it happens sometimes. It happens in this IGFR model. It happens in the Drosophila development model that Stas described. Okay, so finally, a word about dynamics. And this came from when we did the cycle learning algorithm. We tried it on the dynamical systems model. I'm not going to go into it in much detail because I talked about it the last time I was here, although I see now that my audience is not the same audience, but I had assumed that it would be. But my talk, it turns out, is on the web. You can go and look it up if you want to see a little bit more detail on this. But the gist is that we had built this cycles learning algorithm. We wanted to try it on the dynamical systems model. We tried it. We had very nice results. But we still had a few edges that were wrong. And we said, what could be going on here? Why don't we have perfect results, even though we've dealt with the cycles? The CCDs are easy because the data is so well behaved, as in the synthetic case. There were no latent variables. We expected our results to be quite perfect. And so we scratched our heads and we said, wait a minute, that the underlying system here is a dynamical system. And the data that we're looking at is a snapshot in time. What's happening in this dynamical system, and could it be confounding our results? And I'm going to argue that it can. Here's an example. It's not very biological, but it gives some intuition. Let's say I have A, and at time t, it's randomly reset to 0 or 1. B is whatever A was at previous time point. And C is and or a function over A of t and A of t minus 1. What's going to happen is that the correct, I wish I had done fewer builds in this talk, but the correct model is this one, right? Look at this. B is a child of A, and C is a child of A. This is the correct model, the one on the left. Everybody agree? But what are we going to get when we run structured learning? Are we going to get the correct causal model? No. B is going to look like a parent of C, right? Why? Because B is representing A's history, and A is not representing A's history. A is randomly reset, okay? So when I do causal structured learning, I'm going to get a non-causal edge between B and C. Problem. And you might say, well, that's nice, but that's very contrived. You don't have a biological situation like that where every time point, whatever the time point piece is, something is reset. Okay, let's think about a biological example. Let's say I have A affects B and C. And let's say these guys are degraded in the system. Let's say they each have their own phosphatase or what have you. And their degradation system is different kinetics for A, for B, and for C. And maybe A is quicker to turn off, and B is intermediate rate, and C is the slowest. What's going to happen? I argue that what could happen is that you could get, this is your correct causal edge from A to B, you could get an incorrect non-causal edge from B to C. Why? Because B is representing A's history better than A itself. Again, this is a special case of a latent variable, if you think about it. It's just A's history, what A was a little bit of time ago, being represented by one of its children, by B. So here's a little data, little synthetic data that we generated, where we said, okay, well, let's build that system and let's see what happens. And in fact, we do get this confounded model. There are things, though, that made us scratch our head. I see that I'm almost out of time, so I'm just going to kind of.
Can you just clarify something? So the measurement you're doing, right, is A, B, and C at a particular moment in time. So you're not allowed to measure A 2 times ago and measure C.
Exactly. Yeah. So I don't have movies of these cells. If I had movies of these cells, I'd be in a very different domain. But your intuition is asking something. You're saying, well, you've drawn me a time course here, or I've simulated a time course. Well, what's actually happening in a measurement? When I take a snapshot, what's actually happening when I take a snapshot? In fact, how did we simulate those? When we made the dynamical systems model, those were not stochastic differential equations. Those were ODEs. How did we get a bunch of cells? What did we do? What we did was mapping to our biological intuition. This is what we think is happening in biology. What we assume is happening is that the levels of the total proteins are constituting a cell state, and that the cell state is distinct. Tell them I'll get it later. The cell state is distinct from one cell to another. And therefore, when I freeze that reaction at 15 minutes or whatever, 15 minutes is a very typical time point. When I freeze that reaction at 15 minutes, one cell might be here, and one cell might be here. I don't know. And of course, this is very idealized. In reality, it will be very widespread. Different cells are in different places. So in essence, I'm capturing some of the dynamics in a particular time point. Now, people would generally say to me, well, why aren't you in steady state? That's a great question. I'm not convinced that there always is a steady state to go to. Look at these. This happens to be models, but in real data, things look like this as well. Where's the steady state? There's just the degenerate steady state that's uninteresting, where everything is off. And when you try to learn something where everything is off, you're really fighting the signal to noise problem. There is a causality happening, even at this degenerate steady state at 0, because it's not really 0. But good luck. It's really, really hard even when the signal's high. So you might be able to, but it's a hard problem. Sorry. Okay, so the question is, can we avoid dependence on A's history? Again, the intuition is that if I'm just measuring a cell. When I measure here, A is high, but its child C is low. Why? Because the high A here affects the values of C later on. See, there's a delay by the time C goes up. And by the time C is high, A has already gone down. So if I should have made you guys a simulation of this. If you imagine a scatter plot of this data, what will it look like? You may not be able to see the dependence between A and C at all. I think that's an extreme case. I think in most cases you will see a dependence between A and C. My concern is that, and what the model results showed, is that the dependence between B and C will be stronger because B is reflecting that history of A. Okay, and there's a delay in the system. So if you ask yourself, can I avoid dependence on A's history? So we tried, Tanya and I, when we encountered this problem, we said, well, what can we do about this? How do I fix the fact that, if I could make movies of cells, that would be different, but I can't. So how can I fix the fact that I have this dependence on what was before? The cell that I'm looking at is the cell now. What can I do about it? We said, how about if we hold A constant? I'm going to throw an inhibitor into the system. And now, depending on A's history is no longer a problem. If you're here and you're depending on A's history here, not a problem because A's history is constant. So I don't mind being dependent on your history if you're not moving anywhere, if you're constant. And so we formulated an algorithm for reducing non-causal edges. And by the way, this phenomenon of the history of the dynamics confounding things had been described in dynamic models, in CTBNs, by Daphne Kohler. And she called it entanglement, this phenomenon. And so we kept that name. We're calling it entanglement, the confounding effects of the dynamics. And we tried to formulate an algorithm. And in the algorithm I described in the talk when I was here before, you guys can go see it if you want. The idea is to avoid dependence on history by learning from conditions where many inhibitors are applied. We use inhibitors anyway, right? We like inhibitors because they help us understand causality. But in this case, we're using inhibitors in a slightly different way. We're using inhibitors to get rid of entanglement, to hold things constant so that you no longer depend on their history. And so the algorithm was iterative. It said, let's start with 4 inhibitors applied and see what edges we can find. Now let's relax that and take 3 nested inhibitors and add on some more edges. And what we're hoping that this algorithm will do, we wrote a bunch of proofs that I showed in the previous talk. What we're hoping that the algorithm will do is to sway the results, because there's a complexity penalty, right? You're not going to get every possible statistical influence in your model. We're hoping to sway the results towards the edges that are truly causal by having this iterative algorithm that gives more prominence, not more weight, but priority to the edges that are probably not entangled. So we formulated this algorithm, we made a bunch of proofs about it, and we also showed it on that dynamical systems model, and it had nice results. we haven't published it yet, but I'm not going into it. Also, we recently got, I recently discussed it with Joris and some other people, and Joris wondered if some of what I saw was an artifact of the discretization in the multinomials that I use. And I wonder if that's true. And the reason I wonder that is that if you look at, oh, I wish I knew. No, I don't have that. So let's just go back here. So if you look at this graph and you say, well, overall, I think you guys can see what I'm talking about. This cell over here will have high A and low C. And this cell over here will have high C and low A. And then some of them in the middle, they're going to look more concordant. And overall, the scatter plot is not going to show a very strong statistical relationship, and it might not show the conditional independence that exists in the system. So I think overall, you guys can see what I'm talking about. But what if I narrow my range? What if I'm only looking here, or any slice? Something interesting, or an interesting question pops up, which is, well, if I started here, so then I'm just showing an idealized line, but there's actually a big histogram at every point, right? There's a bunch of cells doing a bunch of things. There's lots and lots of these trajectories, and they all have some scatter. If I was a cell that happened to be at the front of my histogram, I was the #1 ranked cell on phospho A, am I going to be the #1 ranked cell here? Overall, we're going to be lower than we were here. But do I keep my ranking? Well, I don't know. It depends on feedback loops and so on. If I was the #1 ranked cell back here with respect to phospho A, is my phospho C going to be the #1 ranked phospho C at a later slice? In some ways, this is asking the question, I'll draw it. In some ways, this is asking the question, if I have an on-off dependence between two proteins, does it look like this? Or does it look like this? Can I see the local correlations? I don't know the answer. In the dynamical systems model, the answer is yes. And so I predict, because of course it's a low noise situation, I predict that, we haven't done this yet because we're working on this other project, but I predict that Joris's prediction here, that the discretization is basically not fine resolution enough. I predict that it will have some effect here. But in a real biological situation, I just don't know the answer. So that remains an open question. Okay, I'm out of time. Do I have anything else to say?
Plenty to chew on.
Oh, so I was showing this is a very sort of cute little example where we said, okay, we trust the combined inhibitors more because we believe that they're less entangled. And so we did a little experiment here where we said, okay, we took a number of inhibitors, I'm forgetting 4 or 5. And in one case, we said, I'm going to have just regular no inhibition, just stim. And then I'm going to have each inhibitor applied separately. 4 or 5, so 5 or 6 conditions total. And here, I'm going to do something different. I'm going to take all 5 inhibitors. I'm going to leave a different one out each time. Okay? So same amount of experiments, but a different approach. And this one is supposedly less entanglement inducing. Does that make sense to people? In one, we combine the inhibition. So each experiment here sees 4 inhibitors with 1 left out. A different 4 each time, and this one only sees 1 at a time. And we predicted that the results will be less entangled when we combine the inhibitors because there's less dependence on history. And in fact, this is exactly the canonical network. And this one has a number of edges that are different, that are not thought to be causal. This is not a simulated data set, it's a real data set, so I don't have ground truth, I don't have a concrete ground truth. But canonically, what's reported in the literature is exactly this. This was exactly correct. So it's evidence that maybe our approach is working in the right direction. And of course, we can think of other approaches. I'm going to end because of time, but we can think of other approaches. And the other approaches are probably going to do something about, can we figure out the time course? Can we learn a dynamical systems model from this? It's not a trivial problem. But you can also ask, as we were discussing before, here's my test tube. What have I captured? Have I captured the entire time course? That's probably too much unless it's a really quick reaction. I don't know. Why don't we measure a timed course? We have to know, in order to know the sampling rate that's necessary, we have to know what's the quickest reaction that's happening. And it might be too quick for us to measure a good time course. T-cell signaling, well, we can observe peaks at 30 seconds, or peaks at 30 seconds. So we may or may not be able to do this, but you can think of approaches that try to get at this idea of, can I capture the time course, and can I use more dynamical methods to try to learn the causal structure? Yeah, so how much is fitting into one snapshot? I freeze the time course at 15 minutes. Do I get cells all along the trajectory? Where are they? Oh, I had a bonus challenge. Should we end, or should I show you guys the bonus challenge?
You've come a long way.
Okay, here's the bonus challenge. So those are my 4 classic challenges, but I have an extra one that's been keeping me up at nights recently. And it is variable noise. It's variable noise. Each of your variables has variable noise. And we alluded to this earlier. We said that if I have an antibody for A and an antibody for B, X and Y, whatever, I don't know much about how much A and B have in the system. I don't know much about how much background binding is happening. How promiscuous is this antibody? It's specific to FOXP4X, but also binds a little bit with a bunch of other things. Just a little. I don't know. I don't know what's going on. I can look at how fat the histogram is, sure. But at the end of the day, maybe that's biological, maybe it's measurement noise. I don't know. So let's look at this situation where I'm depicting a situation here where A is the parent. Let's say this is ground truth. A is the parent, and B and C are both children. Or sorry, B is the child and C is the grandchild. But B is much more noisy than C. This is just synthetic data, it's not real data. And I'm telling you that B has more measurement noise, a lot more. I think I put in 10-fold more in this simulation. So what's going to happen, and I did the simulation just to ask the question, but I think you guys can guess what's going to happen, is that the learned model actually looked like this. C appeared to be a deseparator. Big problem. I mean, in a milder case, you would just get an extra edge from A to C, right? Because B is sort of so noisy that it doesn't really deseparate effectively. But I actually got this happening when I made the noise big enough. I actually got C to be the deseparator.
So C is a better measure of B than B's measure.
Yeah, which is, disturbing. So I think which case you get, whether you can actually get C and B to flip, I think will depend on exactly how bad your noise is and, different things about your system. But the idea is that this variable noise is a real issue. I think, let's consider the milder case where A just affects C. So you have this non-causal edge. You can't get rid of it. It's in the measurement noise. My intuition says that at the limit of infinite data, you'll get the right answer. Because eventually, there is signal there. And, I don't care how big the noise is. If you exceed it with even more data proportionally, is that not true?
The other way. The more the data, the worse you are. But you'll never deseparate A and C from that measure of B.
Well, my assumption was that you can still sort of see the signal of B. It's being obscured by noise, but if you keep measuring, eventually, you'll be able to see that deseparation.
I don't understand. But we can talk about it more.
I don't know. We can also do simulations. This was just a quick and dirty. But never mind the specific results, because it was just a quick and dirty, like, oh, do I still see dependence here or do I not? Do I see deseparation? The bottom line is, that's something to worry about. That's something to really, really worry about. In general, how do we approach this problem? Can we see if my intuition is right, or maybe using some knowledge about this, or some understanding about this, you can say, well, do I have edges that go away when I add a ton of data and are different when I have, I always have a lot of data. In this domain, I always have a lot of data. But is it different when I have 1,000 cells versus 100,000 cells, or even 1,000,000 cells? I don't know. Or can I ask questions like, well, can we start to ask the question of how much measurement noise is in a variable? Can we ask that question? Can we look at variables across different conditions and say, can can we try to ask that question? Can we ask the question, is B ever a skinny histogram? This type of question. If the answer is yes, then we know it's not necessarily measurement noise.
I think one way to diagram this that helps reveal, for me anyway, a lot of what's going on is, let's say A, B, and C is really what's going on biologically. So that's the model of underlying reality, and we believe it. And then you have A prime, B prime, and C prime, which are our measurements of those things. You have arcs from A to A prime, an arc from B to B prime, an arc from C to C prime.
And
you're measuring A prime, B prime, and C prime. But what's really causing the relationships includes the hidden structure of A to B to C. Right. That reveals for me a lot more of what you can expect and what you can anticipate with these kinds of puzzles.
Yes, that's a good way.
And there actually is a solution to it, which is if you have two measures of B, each of which are noisy, but they're separated by B.
What do you mean? Well, how do you have two measures of B?
If I have two measurements of B, two different ways to measure B, that have independent measurement noise,
Oh.
then you can totally solve this problem. This is like,
Occasionally you get that, not always, but occasionally you can get. Honestly, it's quite rare, but occasionally you can get two different antibodies that are binding in different places. A polyclonal would do it.
well, let's talk more offline, but that's a structural equation.
That's a great idea.
But it might depend on linearity, which you don't have. So
The problem is though that you don't know when this is happening, so every variable has to be suspect, and that's a problem. So, but yeah, this is exactly the kind of thing I'm thinking about. How can we formalize this and structure our thoughts around it, given that we are worried about this problem? But I haven't seen any literature on this, at least in this domain. I'm sure there's literature in causality in general, but I haven't seen people concerned about that when they ask causal type questions in signaling. And this is a real issue. We know it's true with respect to our antibodies. Okay, I think that was the last slide. And I think there's an acknowledgement slide coming up. One day.
I mean, sometimes at the moment you get frustrated.
I hit it 20 times. Yeah, so just I want to acknowledge the NOAA lab, which has been amazing and done all the experiments that I've shown, especially Gary. And a lot of this work was done with Solomon Itani, who was at the time with Claire Chomlin, who's now at Berkeley, and a bunch of other people on work that I didn't present. So, and thank you guys for your attention. Even though I went 15 minutes over.
You like to handle your own questions?
Sure. Yeah.
Well, I see this as the leukemia focus side. Are these cancer T cells, or have you done this on normal T cells? Actually, viruses.
You can absolutely ask those types of questions. I personally have not done microbiology work, but there's definitely microbiology. I mean, I've done a little just helping friends analyze the data. There's definitely really exciting data from the lab. I think Drew Hodson from the lab just got a paper accepted in Science Signaling, where what they did there was challenge with typhoid, I think. It was in mice, and the question is asked how. I think he was asking about elite controllers versus non-elite controllers, how their signaling differed in the different components of the immune system. So you can, yeah, absolutely, you can ask these types of questions. One thing that I would love to do is, whether it's cancer or immune, infectious type of attack, I would love to ask the question: what about the network at the immune system level? How is the T helper cell affecting the whatever other type of compartment? And how is the innate compartment affecting the adaptive compartment, and so on? It's a harder question to answer because you don't have the same richness of data. You can't say, within the same cell, I have one data point. In that case, each mouse is a data point, right? And so, or each human is a data point. Because you can't, those statistical inferences are a little harder to make, because you're asking, when I have this number of T helper cells, they're signaling in this way, how many of these other types of cells that are doing something else? Okay, so you have to structure your question a little bit differently, but that's also a really fascinating question. And I think in Drew's paper, He does it in sort of more, just using correlation networks and that kind of thing. But yeah, it's also a really interesting problem.
Can I ask a question about cycle detection? So, I think in many cases in biology, there's homeostatic control mechanisms in place. So if I have my core body temperature and it gets perturbed because air temperature is colder or warmer, right? There's a cycle that happens that will restore.
Feedback loop.
It's a feedback loop that restores. So how do you deal with that kind of situation if it arises? I mean, I know you.
What do you mean? That's what I'm looking for. I'm looking for those feedback loops.
You're looking for it, but if you get a feedback loop put in place like that, you want to observe a dependence even in the beginning. Because if you don't measure on the right time scale, you'll see no correlation between outside air and inside body temperature. You'll look at dependence.
That's really true. So for detecting the cycles, then an activity inhibitor will work. If I see that B is affected by B inhibition, there must be some causal path from B back to B.
That seems.
So detecting the cycles, I'm okay. Check that off. Although you can be concerned that B is just autophosphorylating, and that's kind of boring. But nevertheless, and that happens. It's not even uncommon. But nevertheless, we still detected a cycle technically. It's just not a very interesting one. So we can detect the cycles. But you're saying, well, if you just detect the cycle, if you inhibit B, you're never going to see its downstream children, and that's certainly a problem. It's not that big of a problem because there's all these feedback loops, and so you have all the guys going into B, and then later you perturb somewhere else, and then you'll see B's children as well. So it's not a huge problem. But another thing you could do, another tactic that you could use, although we didn't have to, it worked without doing this, what you could do is turn the system on, wait a little while, and then inhibit B, so that you stop B at a certain point. That's what I depicted, right? I had A start, and then I leveled it off. So you could also do that, but we just didn't need to.
So often when we deal with cycles, stationarity is the assumption that people put onto the IID samples, the distribution. I know you had figures showing that stationarity was not reached, and I'm wondering, is that common across a lot of biological systems?
That's a great question. So I think what's happening is that in the systems that I model, and also in the systems that are being depicted in the synthetic model, the stimulus is a doorbell. It's not a light switch. Okay? And when you have a doorbell stimulus, you're going to respond, and then you're going to go back. Okay? What if you had a light switch stimulus? So in biology, I think you get a lot of both. You get the doorbell, you get the pulse, and you get the steady stim. Okay? And I think when you have the steady stim, well, two things. First of all, sometimes when you have a pulse, you will reach a new steady state. And there are amazing examples for development of this. Do you know Jim Farrell, who does dynamical systems models and small systems? He has a beautiful example. I forget the system. It's in development. And when you pulse the system with whatever thing the development is responding to, it goes into a different attractor state, and it can't go back. Because in order to go back, the variables have to enter a negative domain, but it has to be negative protein concentrations. It's an amazing thing where the development trajectory locks you into a developed state. You can't regress. So that happens, even from a pulse. But in signaling, it probably also happens. I just don't know. But my intuition says, and I haven't checked this, my intuition says that in biology, you have both the pulse and the steady, the light bulb and the doorbell, and I mean the light switch and the doorbell. And my intuition says that probably when you have the steady pulse, the system will go into a different state and stay there for a while. It might become desensitized and then it will go back down. But it's very possible that it will go into a different state and stay there for a while. I don't know, let's say you suddenly go into the sun for a minute. Your DNA damage proteins will turn on, but then they'll turn back off. But what if I suddenly start to live in the sun? All day I'm in the sun. And maybe they will reach a new state where they're active most of the time. I don't know, I'm just guessing. In that case, yes, you will get a steady state. Whether you'll be able to discover all the causal relations from steady state, I genuinely don't know. I don't have a good intuition for this because did you see the cycle that I put up? There were like 4 feedback loops in like 12 molecules. There's all these feedbacks. It's a really complex system. And I just don't know if under such a complex regime, if you'll be able to find the different causal connections. You should, surely you'd find some of them. But I think some of those relationships might be lost. And it just, it's a really interesting question that I don't know the answer to.
I think from listening to your talk that whenever you run Bayesian search algorithm and you get the output, that you expect every edge to have an explanation. Do you allow for random edges to be like false positives, false negatives to happen for random reasons?
Never. No random reasons. All the reasons that I predicted here, yes. Maybe other reasons that I forgot to depict or haven't thought of, definitely. Random, no. And I'll tell you why. If I compute my data, I get nothing. Not one edge. So no, I mean, because these are thousands of data points, you're not going to randomly, I think, yeah, absolutely, your CPD isn't good enough, and so it's not expressing conditional dependence. It's not capturing the sort of leaky dependence, and so you get an edge that's not causal. I don't consider that random. I consider that my CPD isn't good enough. Or all the myriad of reasons that I gave here, where you might get an edge where an edge should not be. And there's probably 20 other reasons, but not random. Try it.
It's a high standard.
Commute the data, you'll get nothing. I got 0.
So when you're dealing with hidden variables, why don't you use methods like FCI like you deal with hidden variables?
That's a great question. So the main reason is because when I was in graduate school, the people I talked to were score-based people. They were not constraint-based people. And I didn't know much about constraint-based, and I'm still no expert. More recent years, I've talked more to the people here than some of the people who came from here and associated people. I frankly don't know which works better, and I am very interested to try the constraint-based methods, especially to couple them with the score-based methods, because there's no reason to have a useful algorithm and not try it, especially when it's still a very difficult problem in the domain. So yeah, the answer is there's no real reason, and I think it's a great idea to try it.
Again,